Hello, everyone, and welcome to today's virtual seminar on flow cytometry assays for cell therapy, which is sponsored by BDOE Biosciences. My name is Christina Pogge. I'm Senior Manager of the Sciences Content Strategy team at Wiley, and I'll be your moderator today. Joining us is Shen-An Wang, PhD. She's Manager at BD for Development Services of Lonza Cell and Gene. Within the next hour, we will learn from Shen-An how Lonza mitigates common challenges in flow cytometry, such as the validation of flow assays. Before we start, I would like to share a few housekeeping notes with you. This broadcast will be recorded and available to watch on demand. You will be notified by email once you can access the recording. After her presentation, Shen-An will be there to answer your questions in a live Q&A session. So please use the chance to ask anything you want to know around flow cytometry in the chat box during or right after the talks. And now I will hand it over to Shen-An.
Thank you so much for the lovely introduction. Today, I will be presenting validating flow cytometry assays for cell therapy. As everybody knows, flow cytometry assay is quite universal for cell therapy programs. It is probably the most used assay, second only to cell count and viability. This is an overview of our agenda. We will look at an introduction on Lonza CGT, which stands for Lonza Cell and Gene Therapy. We will look at the what we actually do in bioassay services, our responsibilities, and what we do to transfer assays from development into GMP. Then we will talk about how at Lonza we use the BD FACS Lyric as a tool to align basically the flow assay across all different sites of cell therapy manufacturing. And lastly, we're going to talk about validation of flow cytometry assays, the challenges we normally face, and some of the considerations that you might want to think about when you're designing, especially flow cytometry assays for cell therapy programs for analysis. Okay. Here is, before we start, this is just the disclaimer slide for Lonza. All right. So Lonza is a global contract manufacturing development organization. We are spread across worldwide, and we have 4 major divisions: biologics, small molecules, capsule health and ingredients, and cell and gene therapy, which is where I am working at. Globally, we have over 17,000 employees. Our history is quite extensive. We have over 125 years of experience. And currently, we have over 35 sites globally around the world. Within cell and gene therapy itself, we have 8 global sites. Within the U.S., Houston is the largest cell and gene manufacturing site, actually probably within the world for cell and gene therapy as a whole. We house both cell therapy manufacturing and also product development services, which includes assays. We have 2 other GMP manufacturing sites, one in Portsmouth for cell therapy and one in Lexington, which is for exosomes. And we have an R&D facility in Rockville, Maryland. We have another cell gene therapy manufacturing site in Europe, in Halle, Netherlands. And we have an R&D research site for exosome in Siena, Italy. In our Asian continent, we have a GMP manufacturing site in Singapore, and we have a small collaboration with Nikon in Tokyo for the Japanese market. So what you'll notice is that we are spread across globally. However, our process development and analytical development actually supports all the manufacturing sites around the globe. Process and analytical development are the foundation for successful commercialization of a new therapy that comes into our organization. What we do is we take the client's idea or a late phase process and move them in a streamlined and efficient manner from the client into GMP manufacturing to ensure that we deliver the medicine in an efficient manner and on time to the patient that is needed. Okay. One of the things that we actually implement globally at Lonza to ensure this ease of transition is our new product introduction and lifecycle process. What this process does is, one, it aligns all new product introduction at any site within Lonza. And the goal is, one, is to de-risk the product introduction into a new site. This is done by standardizing the work streams in 6 key areas. This includes analytics and also manufacturing. This in turn gives a result where we have a well-defined process that is executable repeatedly and consistently in manufacturing for GMP release. The bioassay services is the analytical development portion of for cell and gene therapy. We own the entire analytical life cycle from early phase and preclinical all the way into commercial and post-commercial. We are considered the technical subject matter experts for all non-compendial assays. So what are some of our responsibilities? We are responsible for development optimization of any assay that are in-house from the clients. This can be new assays, and if the client do not have an assay and they need us to develop one, we can develop one from scratch using our wide and vast areas of expertise, because we have encountered so many and developed so many assays for the various different cell therapy programs. We are also responsible for drafting and finalizing the GMP analytical protocols and the continued refinement of these analytical protocols and methods in close collaboration with QC as the assay moves through its natural life cycle. We are also responsible for the assay technology transfers, and we are responsible for this. Either we transfer within Lonza, because we sometimes do have programs that will go into more than one manufacturing site, or we can transfer out of Lonza as a client if they want to go either have a second CDMO because capacity and other issues, or we transfer into Lonza. So we can do it both ways. We are also responsible, and we also own the assay validation process. Bioassay services designs the protocol and a strategy for validation of all the non-compendial assays. And we are quite well versed in this, and we are also very up to date in our regulatory understanding of the guidelines for assay validation and also some of the release requirements for the cell therapy programs. Okay. Due to our global presence and obviously the ever-changing landscape of cell therapeutics, we must remain flexible in basically our manufacturing capabilities. Now, one of the ways is obviously our global presence, but this also means that we have to ensure that our manufacturing sites are aligned across the globe, that each manufacturing site will function exactly like the other, and that there are no inconsistencies that we cannot correct. So obviously, one of the biggest challenges in ensuring consistency is assay alignment and assay consistent performance from site to site. It's one of the ways that we ensure consistency is obviously in aligning the equipment, which is actually one of the central part of what we do. When we in-housing equipment, we look at to make sure that it has the features to support the 2 compliance markets in the US and also in Europe. And in addition, we also make sure the instruments as it comes into Lonza is able to comply with the Lonza validation process, where we actually validate the instrument for GMP use. The next is ensuring that our assay validation protocols and executions are consistent from site to site. Obviously, regulation guidelines are up to interpretation, and sometimes different people can have different interpretations on how to design protocol for certain parameters. This can lead to inconsistent assay validation processes and strategies from site to site. what we have started doing is basically start aligning these protocols for various specific assay groups so that we're looking at parameters in the exact same way for different assays at different sites. The last thing is actually ensuring the consistency of how we report the GMP data. And this actually is aligning how our assay reports are generated, how our documents are generated, and how we track and document our assay executions at different sites. I'm going to go into a little about the flow cytometry assays. Like I said, it is nearly universal for cell therapy programs. From my experience, almost every cell therapy, and I have to say almost because almost every cell therapy have at least one, and most have 2-3 different types of flow panels for their program. And I'm just giving you an example of sort of the 4 major categories, and this is not inclusive, of what flow cytometries are generally used for in cell therapy manufacturing. The first group is basically assay for identity and phenotype. The phenotype assays are mostly in process. They basically look at the major leukocyte markers that most people would look at. The identification assays are usually release assays, where basically it is the identification release criteria for the cell therapy product. There's also assays that are implemented for purity and or impurities, and a lot of these assays, some are on the major leukocyte markers that resemble the phenotype, but some can be customer-specific targets to look at the purity of the product to set release criteria to release the product for clinical or commercial. The last 2 categories are actually normally not part of a release criteria for the product, but they are nevertheless quite important and often used. One has to do with dose calculation. We do see often, and our experience with, for example, in CAR T cell therapy, dose calculation is often a combination of the flow assay data in conjunction with a cell count and viability data to give a dose calculation of the therapy itself. The last category is characterization. These assays are normally for information only. They normally do not have a specification or any release criteria. They're often used in clinical, and some programs will also use them in commercial to just look at characterization of the product as it goes through the process and at the end of the process. A lot of these, for example, if you were talking about CAR T cell therapy, T cell exhaustion panels, apoptotic panels, and memory panels are often characterization assays that a lot of people would like to use just to look at how their T cells as they go through the process and how they respond. Okay. Now, due to the critical nature of this flow cytometry assay for cell therapy, one that we have to do is align the flow cytometer across different sites. And one method and one process that we have implemented is basically aligning the flow instruments. And one of the flow instruments that we have implemented at all cell therapy manufacturing sites, and this is in the quality control lab at Lonza, is the BD-FACS Lyric. Currently, all 4 manufacturing sites have the BD-FACS Lyric implemented and validated and released for GMP use in quality control. We also, as bioassay services, we're the one that develop and optimizes the assay. This includes, we have normalized and standardized how we built the GMP templates for these flow assays. And also we have standardized how we design the reports for these assays so that in GMP, all flow assay have very standardized report structure for data reporting. And One of the advantages that we have seen with the BD Fast Lyric is that it has a function to enable a seamless assay transfer from instrument to instrument that is quite critical and is quite useful for GMP execution of flow assays. Okay. So what do we mean? What I mean about ensuring assay consistency? So one of the first thing, obviously, that we have to do is ensure consistency, not just between GMP to GMP site, but also between development and as we transfer the assay into GMP. And one of the ways to do that is in how we actually set up the instrument. The instrument setup for the BD Fast Lyric, there are two options, default and user-defined settings. At Lonza, due to the nature of our business, we mostly uses the user-defined setting, as a lot of our assays are client-specific per the program that we are supporting. And in order to ensure that we can transfer these setting options in the assay from instrument to instrument, there are two options that we actually use. One is because we do have more than one development site, we have to be able to transfer development protocols from site to site. And in that case, when we transfer development protocols, we transfer the experiment. This way, the other site can use what we have and build new assays on top of that as a foundation where everything in the assay is editable. However, when we transfer into GMP operations, for example, like the QC labs, the assay transfer is actually in workless mode. In this mode, the assay transferred cannot be edited, meaning they cannot do anything, edit the template, they cannot adjust the change the hierarchy, and they also, one of the main thing is the report structure is also transferring workless mode. And this is an example on the right of sort of what how we standardize our reporting for the flow assays. A lot of these requirements are to ensure that we have good data integrity and audit trail tracking. But one other advantage is by standardizing report, you standardize all of the plots that you want to show so that no report have different plots, or you forget something on one and then you're missing information. So standardizing report structure allows us to make sure everything comes out consistently every single time in GMP execution. And how we actually do transfer the assay, we've basically used the BD application note that has some instructions on this. And if you need to look into it, this is actually available on the BD website. So for the next few slides, we are going to look at some sort of sample data that demonstrates how the assays as you transfer instrument lab to lab remain consistent, looking at some of the example panels that I'm going to be showing. So the first set of example data is looking at instrument to instrument, and this is the instrument in PD to the instrument in a QC lab. One major difference between the instruments between development and quality or instruments you use for GMP executions is that in the quality lab, these instruments go through a validation process where they are released for GMP use. PD instruments do not go through this process. And what this demonstrates is that our validation process has absolutely no impact to the assay itself and to how basically the assay performs on the BD Fast Lyric. So for this one, we use a commercial control, which is a lyophilized human PBNCs. The other thing I want to make sure is that that I make a mention that the lots of this control is matched between these two instruments, hence why the CD3, CD45 positive population are actually quite similar. What is important is also you will notice that the plots are very consistent across these two instruments. Most importantly, the quadrant gating are very similar, if not exactly the same, between these two instruments. That's ensuring that the assay perform and the assay perform consistent from instrument to instrument. Our next example is looking at basically transfer instrument at the assay between instruments within the QC lab. And these are assay transfers from a GMP release instrument to another GMP release instrument. This is critical to ensure consistent performance of multiple instruments in QC due to the nature of our business. When we execute assays for release of GMP batches, we do not isolate, let's say, one instrument or another for one assay or the other. We allows the freedom, and also for GMP, you have to have multiple instruments to ensure that you have backups so that your assay can be executed in a timely manner. For this one, what we notice is that using the same commercial control, lifeless human PBMCs, against the lot match between these two instruments, looking at the same CD3 panel, we notice again that the CD3 percentages are fairly consistent between these two instruments. And in addition, the quadrant plots, the gates are drawn at nearly the same fluorescent intensity between the two instruments. Our next set of data is basically demonstrating how assay consistency is demonstrated irregardless of your positive population that you're looking at. So this is now looking again on between two QC instruments. We are using a different control, and this is a commercial control that resembles sort of a human blood sample. So you will notice that the CD3 population here is dramatically lower than the previous slide. And in this case, even though our positive target population that we're interested in and our reportable data is around 30%, between these two instruments, again, the data is fairly consistent. But most important, the gating is very consistent between instrument to instrument. Next, we are looking at the same commercial control, looking at CD19 positive population cells, which has even lower event counts within this sample compared to CD3 positive populations. And in this case, we see that even though this control has CD19 around 5 to 6%, the data generated from these two instruments are fairly consistent, even at 5 to 6%. In addition, the gating for this is also very consistent from instrument to instrument. The last set of data I want to exhibit is when we transfer an assay from one site to another. And this is a comparison between the data that would generate at the Houston site, where the Houston site transferred an assay to the site in Heilongjiang, which is in the Netherlands. And for this one, we use a commercial control that it's called a CD34 PBMC. One thing I do need to mention is because this was executed at completely different continents, we could not ensure that the control lots match. So the lots for this are not matched. So what you will notice is due to that, the CD34 percentage reportable data is not exactly the same as the previous examples. But what is most important here is that the positive gate around the CD34 is consistent between the two instruments, even though they're at different sites. In addition, you'll also notice that the range of the fluorescence intensity for the negative and the positive population are also very consistent from instrument to instrument. So now that we have implemented the tools to keep assay performance consistent from site to site in our GMP manufacturing sites, what I'm going to talk about next is we're going to move on to some of the challenges that we often encounter when we try to validate flow assays. As everyone knows, an assay, in order to be used in the GMP setting, has to be validated according to regulatory guidelines, looking at different parameters that we do assess. And we're also going to talk about sort of some of the considerations that when you set up new flow assays that will eventually go into GMP to think about as you're setting up these. Okay. So Lonza has a sort of a phase approach. It's called a phase appropriate approach to assay validation. This means that we have a approach to get your assay ready for clinical production, and then we have a separate process and design a strategy to get your assay ready for PPQ and commercial. And for PPQ and commercial, basically you're ready to file your BLA and to get your drug license by the regulatory agency. Now, there are some differences, but when you design your flow assay, some of the things you have to keep in mind is the control selection, because your control selection will also play into your validation of your assay. Now, there are normally two different types of controls for your flow assay at the minimum. One is a sample control. These are controls to demonstrate that the sample when it's executed is valid. And this is mostly your gating controls, such as, and you can choose what will actually be appropriate for the assay that you're developing or getting formalized. So these controls are things like unstained, FMO, isotype, which we don't see used as much, but there are still some assays that will use an isotype control. And for us, we also do event count as one of the controls for the sample to ensure that enough events are counted per tube to make the assay valid. The second is the assay control. These controls are to ensure that your execution of the assay is valid, meaning that there's no issues during execution that could cause an impact. And these assay controls, again, there are quite a lot of varieties to choose from, and you have to choose the one that are most appropriate for the assay that you are developing. These controls, there are quite a few commercial controls with a COA with ranges for different markers that you may be looking at. We also use a lot of product controls. These product controls are usually reserved for flow assays where the target is program specific. So, for example, a lot of the CAR T cell have mostly use a product control because the target against that CAR is program specific, and a lot of these do have customized antibodies that is developed for that specific purpose. And then the other option is looking at positive negative controls, and these normally talk about are based on cell lines. Again, you have to look at what is most appropriate and fit for purpose to be implemented for your assay. Now, one of the areas that may impact assay performance is actually sample stability. And the reason that this can have an impact is due to the time it takes to basically process and stain a sample for flow cytometry. And this is sample stability. I mean, when I was in academia, it's not something I consciously think of. But in GMP executions, it is actually something that is quite critical that you should consider when you design your flow assay, especially the protocol. And what can actually impact sample stability? And what we're talking about here is basically from the time the sample comes from manufacturing or from receiving to the time that sample gets being acquired on the flow cytometer. And for routine testing, the panels that we currently execute in GMP you can look at anything from 1 to 6 hours to reportable data. And when I say reportable data is because when we for qualified assays and validate assay, the reportable data have to be released. So there are review processes. So there inside there is quite a time difference. For assay validation, it has to mostly do with what sample preparation we have to do in order to assess the different parameters that we do for validation purposes. Some of the two major parameters that we do is precision and accuracy. Accuracy is also part of linearity. Precision, normally we can just do the routine assay executions. So the time from receipt of the sample to acquisition of the flow cytometer and reportable data can be 1 to 6 hours. It's not much different. However, as if anybody has ever looked and did validation of flow assays for accuracy and linearity, the sample preparation for these samples can be quite extensive and quite lengthy. And because of the sample prep time for these, there is quite a time difference from the time that we receive the sample to this time when the tubes go onto the flow cytometer for acquisition. And it can be from anywhere from 6 to 12 hours, depending on the size of the panel and the complexity of the validation protocol. The second major thing that can impact your sample stability is actually the sample itself, your sample type. Now, in cell therapy, because of the starting materials, especially for auto cell therapy, comes from from patient material or individuals. The leukopak is one of the most common starting material that we see. The processing time, if you're working with a leukopak, it can actually have sample stability issues if the leukopak sample has any sort of delays. One is because the leukopak has a reversible anticoagulant, obviously for the apheresis, it can cause issues if you over dilute the sample and you don't realize it before proper processing that can actually start activating your coagulation factors. So one of the things that you should consider in your protocol is how to clear out any potential of coagulation happening so that way you don't lose your sample because the coagulation activation, and then it will have an impact to the assay. The second one is actually stability of the lysis buffer. We have now seen quite a bit where the protocols, the lysis of red blood cells for these samples are at the very end after staining, and we actually acquire the sample in the lysis buffer itself. For these, you have to evaluate how long your sample is stable in that lysis buffer prior to acquisition, because obviously lysis buffer can have an impact on your sample the longer it sits, irregardless at what temperature. The last two sample type that we're going to talk about is the fresh sample and the cryopreserved sample. The fresh sample can include the leukopak, but it can also mixed cell populations, but it can also include the purified cell populations. Obviously, purified mixed populations have different sample stability considerations that it is highly recommended to examine when you're developing your flow assay for these samples. Cryopreserved samples, obviously, the main issue is the cryopreservative in the that's in the sample that has to be cleared up before you can run your assay. Normally, it is not that much an issue. However, this is still something that has to be considered as you're developing the assay for these type of samples. So next, we're going to talk about what is the Lonza approach for how to evaluate and assess some of these parameters that we normally see very commonly in flow assays. One is obviously precision. Now, precision is defined as a closeness of the data, and I have a very simplified Bose-Einstein picture here to give everybody an idea of what precision of data means. So basically, precision of data is basically how close the data are to each other. For these, when we get an assay ready for clinical production, we always look at intra-assay, which is just repeatability, precision, and also intermediate precision. But for us, because this is for clinical readiness and we want to make sure that the assay is ready for clinical production usage, we try not to bring in potential sample variation that could impact the assay. So for these, because we're mostly focused on precision of the assay itself, we normally try to make sure that the sample that we're using for validation is quite homogeneous. Now, when we get the assay ready for PPQ and commercial, if your assay is being executed at more than one site, even more than one lab, reproducibility has to be considered. And this is basically lab-to-lab comparison and precision between the labs. But one additional variation that may have an impact that we almost always look at for these late phase clinical studies and commercial assay readiness is lot-to-lot variation. And most of this is probably most appropriate for autologous cell therapy or allo cell therapy where the starting material comes from an actual individual. Obviously, that will give you a lot more variation in your sample that may impact the precision of your assay, and that should always be as part of the consideration when you get this assay ready for commercial and PPQ. The next set of parameters that we want to talk about is accuracy. Accuracy is defined as how close the data is to a reference sample or a reference value. For flow assays, as we all know, there is no true reference available. So a lot of times what we do and how we do the accuracy assessment is by percent recovery to a known value. Now, for clinical readiness, what we normally do is to assess the marker in combination. I will give you an example what this actually means. And how we do the reportable data usually follows the routine assay, unless our hierarchy doesn't allow it. Now, one of the reasons why we talk about markers in combination for clinical readiness has to do with flow panels that we all know. Often has more than one marker. And when you're trying to do accuracy, it can't affect any and all the markers within the panel. So to keep the assay for clinical readiness, we just want to make sure that the panel is performing as intended for clinical release of clinical batches. For these, this is what I'm going to show you how sort of the approach that we have been taking. We use the 100% of the target is always sort of our reference. And then we do percentage. You normally dilutions are percentage down from the 100%. And we always use the highest marker in the hierarchy. So for example, if you're looking at a panel that has 45, 3, 4, and 8, your 45 is always reported out of your singlets and or live cells, depending on how the hierarchy is designed. And so, for example, if your 100% of 45 is 60%, that means at 20%, your CD45 will be dropping to 12%. And for this one, you can look at accuracy of CD45 by reporting from the parent gate. Now, there is one caveat because if we do all these markers in assessing combination, if you have CD3, 4, and 8 that are reported out of the parent gate, so that means 45 is out of 45 for CD3 and out of CD3 for CD4 and 8. You have to be careful because these percentage will not change because the only thing we're changing is the entire sample altogether. So because these percentage are reported directly out of the parent gate and only CD45 is moving, if you keep this same reporting structure, you will not see these percentages go down for these markers. You actually have to move them out of this parent gate into the SIGA and live cells, because that is how the assay validation protocol is designed for these purposes. This is where you will see it. And for us, this is sort of a way to get a flow assay validated in an efficient manner for clinical production. Now, when we actually get a flow assay validated for commercial readiness and PPQ, we actually do assess all markers separately. The strategy for this is to ensure and to demonstrate that each marker of the report of the data in the flow panel are not dependent or somehow linked to another marker. Because if it does, you may have other considerations you will have to put in. Now, granted, a lot of times your panel will have reportable data and also FIL markers in the same panel. The FIL markers may be omitted from being validated separately. This can be discussed with your regulatory personnel when we start looking at assay validation for commercial readiness. So what does this mean actually? This means that if, for example, we are trying to do accuracy on CD3, CD3 will be out of CD45. And in this case, because CD3 at the 100% is quite low, around 30%, you will have to actually do accuracy samples that goes above 100% and also below, meaning that you have to increase only CD3 without increasing any of the other markers, and also decrease CD3 without decreasing any of the other markers. And Alonzo, we have done quite a bit of this so that we are able to control variation only in the expression of CD3 and when it's reported out of CD45 gate. So for a 150 sample, CD3 percentage should be 45, and at 20, CD3 percentage should be 6%, and nothing else in any of the R markers will change. One of the most difficult markers to actually assess for linearity accuracy separately is the live and dead. And we have done some studies looking at how to kill a specific sample. So the live cells and dead cell population will vary without affecting any of the other markers in the panel. And what I'm going to show here is to demonstrate that we can use ethanol, and we investigate the ethanol and the fixation and permanentization buffer to prep the dead cells. And the only thing I want everybody to take away from this is that how you kill your dead sample may be dependent by your sample type. So for the Lucas sample, this is the data that have been gathered when we do ethanol and fixation and permanentization. And you will notice that when we do the same thing with the purest fast sample, we do actually get slightly different data. So when you design your validation protocol for these type of assessments, you have to do some pre-validation work to determine which is the best protocol to actually use in your validation. Not our validation protocols will work for every assay or every sample. So in summary, the analytical direction at Lonza for cell and gene therapy is moving basically towards standardization at all CGT manufacturing sites. One way is ensuring that the same flow cytometer is implemented at all sites, and one of them being the BD-FACS Lyric is currently installed at all CGT sites. We are also making sure that all the BD-FACS suite, which is software that runs the BD-FACS Lyric, is all on the same version at all the sites. And also we are doing the assay transfer using the BD-FACS Lyric protocols to ensure that the assay transfer is consistent from instrument to instrument and site to site. As part of our consistency and trying to standardize everything, we're also looking to standardize the flow assay protocols, and these are the routine assays. In this way, we are aligning how we do the flow templates for every single flow assay, making sure we have all the required data that's that is needed. And also we are aligning on the sample and assay controls that have to be implemented for each assay. Lastly, as part of the standardization, we cannot forget about our strategy to assay validation. We are standardizing how we design the protocol and approach to how we assess each parameter for assay validation, and also standardizing how we define the minimum criteria for each parameter. And with that, I will take any questions.
Yeah, thank you so much, Shen-An, for this very interesting presentation with all these useful tips and application examples. Before we continue with our Q&A session, this is just a quick reminder to our audience that we can still take your questions, but we will start with the ones that already came in. So, can you balance or same instruments with different optical layout?
So when we actually do that, that's actually it's actually called a transfer of an assay from one instrument to another. So one thing to keep in mind, assay validation is actually instrument specific and also sample specific. So if you move an assay from one flow cytometer to another, and we're talking about completely different type or a completely different model, the first thing you actually do is have to assess comparability. And we actually do some feasibility and comparability studies to demonstrate that the data is comparable. And then after the transfer, when the assay is set up in the new cytometer or whatever you want to use, then actually you have to design a revalidation protocol to look at the parameters that are most important when you did that transfer. So it is no in the simple sense, but it is also doable because you just have to have built in a track record of data that will support the transfer.
Okay, thank you. There's another question on validation. So how do you define tolerances to accept validation between two different instruments?
So when you look at comparable data between two flow instruments, or actually any comparable instruments of any sorts, we actually do a equivalence analysis. And I don't know how many people, it's a statistical analysis that you can actually do to look at how equivalent the reportable data is between the two instruments. So far, we have noticed that at least for flow cytometry and flow cytometers, the changes can be mitigated if there are major differences, but so far we have not seen any.
Okay, great. So you presented different protocols, and there's one question that's what is the rationale using isotope controls for sample controls when it is understood that they should not be used to determine or establish positivity in fully stained samples? Additionally, the same protocol you provided utilizes FMOs.
So that's actually just an example of what could be used. And I do have to say, because we are a customer-centric organization, we are client, we basically serve our clients. We get a lot of assays when they come in where the client has very specific protocols that they want to use. They have very specific controls that they want to use due to various reasons. So while if we do design the assay in-house, we normally do not we do not use the isotype as a sample control. This normally only happens when our client has a protocol that's set using that and they do not want to change it, or they have a very specific reason for using it. So that is correct. This is more of a preference of our clients when they transfer the assays in-house to us.
Okay, makes sense. So another user would like to know about certifications. For example, if a CRO lab that run flow cytometry supporting flow panels supporting clinical trials for cell therapy and specific certifications. For example, GMP and or CLIA.
Right. So for us, our quality control, because we are a GMP manufacturing site, we are basically under the GMP requirements. However, we do also outsource a lot of our assays because depending on the program, some of the assays that are required for like compendial executions, things like that, those labs, outsourced labs, that I assume has their proper certification. So for us at Lonza, because we are GMP, we operate under the GMP guidelines.
Perfect. Thank you very much. So the percentages of the CD4, CD8 will fluctuate in different disease conditions. How does the validation work then? Will you run the normal samples in validation?
So yes. So for cell therapy, auto cell therapy programs, we almost never use an actual patient sample to validate your assay. There are several reasons for it. One is an amount of sample that actually take to validate the assay because of the different parameters we have to assess. The other is nobody really thinks that we should, exhaust the patient sample, which can go back to the patient to basically use it for validation purposes. So a lot of times how this happens is that they will generate a represented drug product from using a healthy donor apheresis material, leukopak, or a healthy donor type of material. That being said, not every assay, flow cytometry assay, yes, can normally be executed on normal donor material. However, that may not be true, especially if we look at flow assays that may have designed to do potency readouts. So for those types of flow assays, it may be required, it may not be possible to be executed on healthy donor material, and you may have to use a clinical samples. But for those, we always limit the samples that we take for validation activities, and basically we'll do a combination of healthy donor versus clinical material to ensure that whatever amount of material we pull out for validation purposes is still able to generate the dose required so that the product can go back into the patients.
All right. So can you tell us a little bit more about Lonza's process to transfer flow assays developed on a different flow cytometer? Is there a difference in the assay being transferred, if the assay is being transferred that is qualified or not?
Yes. So for our first criteria, we always advise our clients, especially if they transfer flow assay into us, is that we have certain cytometers that are validated for GMP, and ideally we should use what we have on site that's already validated. And the process that we will go to is we will always do a feasibility and basically set up the assay, and then also do a non-GMP comparability just to get some data that we can use. And then we basically revalidate the assay on the flow cytometer at the site of GMP manufacturing. Now, it is up to the client whether they want to switch to what we have available here, or if they want to in-house their own instrument, which is also possible. There are timeline considerations because whatever new instrument we take in-house, we have to release it for GMP. So that does have some extended time to get the instrument GMP ready. And so the process is usually development, then qualification.
Okay, great. One user is wondering if you know if there are any guidelines available for running controls and protocols.
So not really. So if anybody has ever seen the regulatory guidelines, the ICH, even the FDA guidelines, which actually follows a lot of the ICH guidelines, and even the ones from the EMA, they're all kind of vague. They tell you that you have to have controls, but they don't specify exactly what type of controls. So the normal ideas is, and it also depends on the assay. So for flow cytometry, you almost always have a sample control, meaning that it ensures that the sample is basically adequate to, is valid to be used for reportable data. The other one is the assay control. The assay control is basically a control that goes through the entire part of your assay process. And it basically demonstrates that you have stained everything correctly, all the markers are expected to where they are, et cetera, et cetera. So those are usually the, I would say, the type of things in general that you will have to look at. And this is across all GMP assays, not just the flow. And for the sample controls, as part of flow that I mentioned before, one of the other things that you also have to look at is the gating controls, which is part of the sample controls.
Okay. I have a very quick question, I guess. Does using ethanol to kill cells add to autofluorescence?
So far, the data that we have gotten doesn't really notice, extensive autofluorescence issue that will cause an impact to the assay. That is not to say that it can't, depending on the panel that you're actually assessing, and also the live dead channel that you're using to do your live dead gating. But the ones that we normally use so far have not seen too much of an issue.
Okay, great. Is there any advice you can give to startups and biotechs what they can do early on to make flow cytometry assay transfers easier later down the line?
One is identify proper controls. Ensure that you have the control that you will need for each assay. Ensure that your gating strategy and this is not just talking about hierarchy and how the data is reported, but also where you set the gates that they can be as objective as possible. We have noticed that if you try to, I know for people who are very experts on flow and stuff, that we do a lot of gating by visualization. In GMP, this doesn't work as well, because if you, as everybody knows, everybody will have a different idea of what is the middle of something, what is positive, what is negative. So having as objective of a instruction for gating is very critical, actually. And then also making sure that you have the proper controls, and that will help quite a bit.
Okay, thank you. And do you recommend compensation to be done every day, or do you establish compensation stability and do compensation like every month, also depending upon the established stability?
Correct. That kind of depends on the flow cytometer, but for the FACS Lyric, we do establish, because our panels are usually user-defined protocols, so we do establish the stability of that compensation normally during development or running when we do feasibility. So we have data that's generated consistently. So we actually just go with the recommendation that BD has for the BD FACS Lyric, which is the 60-day.
Okay, great. So I think we can do one last question, longer one. What are your thoughts on flow assays like T-cell memory phenotyping, where there is no gold standard for the markers being used for T-cell phenotyping? Do you have any orthogonal assays to confirm the memory phenotypes?
So the easy answer to that is there is no standard. You are correct, and most of these assays are never used for release. So you can actually use whatever panel that you feel and whatever marker that works best with the product that you're analyzing. I have never so far seen anybody use a memory phenotype panel to set a release criteria. They mostly just monitor the cell product through the process, and then at the end to see what it is. And then my assumption is that they may use this data when they do the clinical trials to see if there's a correlation. But so far, I have not seen any program that actually uses this panel for anything but just for information only.
All right. Thank you so much, Shannon. Again, thank you for your presentation. We have to wrap up the Q&A session now. Thanks for everyone who attended, who asked their questions. And yeah, Shannon, I think it was really useful, your presentation and all the questions you managed to answer in such a short time. So thanks again, and everyone have a great rest of your day.
Thank you very much.